I want to first express my appreciation and thanks for the ILSI and the sponsors of the companies who support ILSI for sending the invitation to me. I really thank you for this. It's a great honor to be here in this august audience. And my job, before I proceed my real talk, I had to do something to make all of you alert after the lunch. In America, we have a tradition, because since I lived in the US for 40 years, so I know only the American traditions. Before the football, baseball, they have a session called a warm-up sessions. How many of you seen warm-up sessions? Well, not many. You know, I know, Richard, you know that. And also, the same thing we in India copied on the T20. I'm sure you must have seen T20, right? So there we get actually not only the warm-up person from India, we actually import warm-up girls from USA for this. Okay. So after some time, the USA ran out of some pretty girls or whatever. They started getting old people. One of them, some of you may know, his name is George Burns. Anybody raise your hand? George Burns. So George Burns was actually 94 years. Then he actually, in London, they actually planned his 100th birthday celebration. So essentially now, for whatever reason, they brought me here. I'm one of those George Burns of toxicology in India. Thank you. With that intro, just so that all can smile, I will move on to my first slide. Okay, this talk is kind of a very general. I kept it because I want to give a primer for the future. Other speakers, 3-4 speakers are there, which will go in depth of the toxicology and risk assessment part of it. Essentially, it will cover some of the things that I've gone through, but I capsulize in such a way. I have only 15 slides, not many, so I'll cover it easily. So in 20 minutes that I have. So essentially give you a broad outline to focus for you, in addition to the microbes, which you covered quite a bit, as to what is the source of the foods. And food is a fundamental importance to all of life. Human, this is something astonishing. When I was working for a Searle company in USA, I developed the aspartame. So at that time, I was searching a lot of these things. The average human being consumes 30 tons of food in his or her lifetime. That's a phenomenal amount. So the amount of contaminants that Dr. Kanungo mentioned is, again, we'll be accumulating, absorbing. At the same time, I don't want to scare you, my goodness, all these things are killing us. No, because all of your life expectancy has practically doubled in the last 25, 30 years. So essentially, all of you here are going to live at least 90 years, the current younger generation. So essentially, life expectancy is going up. So essentially, these things, contaminants is a problem, no question. All of those things which I'll talk. So food obviously is a mixture of nothing but chemicals. Water is chemicals, food is chemicals, body is chemicals. So it consists of predominantly 99.9%. You are talking practically 100%, nothing but nutrients. Proteins, carbohydrates, fats, vitamins, minerals. So contaminants, we're talking a very, very small quantity, toxins, contaminant additives, which I will talk to in the next slide. Okay, now let's start from the example. Where do they come from? Obviously, we are exposed to 3 types of things. One is crops, livestock, seafood, and obviously air and water. And the contaminants actually do come from these things we talked about, from vehicles, agriculture, waste, industrial emissions. These are a part of the modern life. None of us can afford to live without those things. Otherwise, we'll be the boonies. Okay? So essentially, they are a part of us, and we are a part of them. We need to sort of learn and find out how we can harness this existing good technology and manage it properly, where the exposure of all these things can be contained and managed. That's very important. Now, once they come into the food chain, obviously, then nothing is we are eating straight away. Everything is processed. So processing and storage and distribution is an issue where there is a possibility of exposure of extraneous materials which are not indigenous to food. So after having processed, service, distribution, it comes to the retail. From the retail, obviously, it's not the end of it. After the retail, practically most of the stuff that we talked earlier, even milk is cooked, boiled, other things, we bring it and cook it here. So during that process, there is always not only a destruction of the contaminants and pesticides, there is a new generation of chemicals can be generated the way depending upon how we cook. So all these things come into the picture. I'll give you some series of pictures of Indian food rather than beef and other things. we'll see that. So humans and animals, we ourselves are a great reservoirs of environmental sources that actually we are exposed to. Very important. Now, a little bit background. Typical normal diet contains hundreds of thousands of compounds, and also some of them are in situ when food is cooked or prepared for consumption. Safety of food cannot be proved. It's very important. I don't want to go into the illusion that food is safe. Safety of any food cannot be determined because the food itself cannot be given to any animals. If you take any food that you eat and we give to rats, mice, or dogs, or monkeys, you are actually destroying the nutrient composition of that. So essentially, there is a limitation for that. So of course, any food additive or anything that we add on to that for whatever the definite functional purposes, or a contaminant that becomes a part of the food, certainly they need to be tested. And let me tell you, across the world, both in the United States, Europe, Japan, Canada, Australia, all these countries I visited, lived there in all these countries, they have very good mechanisms. And fortunately, India's one good thing is they actually take advantage of the existing systems in place around the world. They don't want to duplicate that exists there. And certainly, over and on top of that, they will do their own assessment and then approve the products. So the regulatory framework was very important. This sentence is last is very important. The regulatory framework works on the presumption of safety. As in, it's always safe unless it contains a known harmful substance amount likely to harm. It may be there. Minute quantities of contaminants are going to be there. They are what is called as the de minimis, minimal risk. We don't have to worry on that. Now, if there is an ingredient that is added to food by design, we know hundreds of them are added there, which I'll come back to that, or a contaminant that comes, which Dr. Kanungo alluded to, because he's from the pesticides, he knows these do come into the foods. They demand a higher standard of safety because they are a part of our food, which is added for various functional purposes or industrial things come up. So safety concerns with added substance focus on the nature of the substance and its intended conditions of use. I want all of us to understand, particularly working in the food industry here, it is the intended use and intended exposure, intended duration of exposure, intended amount of exposure. So the exposure is the single dominating factor in all these food assessments you need to. Toxicity, we'll talk about it. Toxicity is innate property of every chemical. So as a result, don't concentrate only on the toxicity. I'm not telling you to ignore that. That's important. But in this food business, the exposure, the duration, the population, all those things are very, very important. So specifications for added substances now depend on the concept of a threshold. This concept is very, very important. Every chemical, no matter what, including the dioxin, I hate to say this, there is a finite amount of that particular chemical that a body can handle. Bodies are made up of nothing but enzymes which have an ability to destroy the exogenous or xenobiotic compounds, including pesticides, contaminants. So at the same time, just because body can metabolize the handle, it doesn't mean we need to take all the junk. Absolutely not. So essentially, minute quantities of these contaminants are known amounts of the food additives that are intended for a functional purpose. They are, after having tested, the levels are set. This is called a threshold level. The basic concept here is this is the concept that is developed by the U.S. Food and Drug Administration in the U.S., which is called the de minimis. There is a minimal risk. So again, they have come up with a 10 to the 4, 10 to the 5, or 10 to the 6, depending upon the nature of the effects produced. So at a lower level, which is not worth worrying about. Okay, now let's come to the food toxicology proper. Substances found in food that might be harmful to those consumed sufficient quantities containing such substances, which some of them Dr. Kanungo already alluded in his introductory comments. Inherent toxicants that are present in the food naturally, which I'll have some examples. Naturally found in foods, contaminants which are in preparation, and processed foods. Very important. Don't forget processing of the food. So these are some examples here. Here, naturally occurring ones, food constituents like carbohydrates, fats, proteins, these are natural constituents. Natural toxins, I'm sure I don't have to teach here, aflatoxin was very, very well known since 1961, which used to be called turkey X disease, when 100,000 turkeys died in England, and that's when the whole thing started. Now we know a lot more than aflatoxin, other mycotoxins. And other chemicals in foods that are there, like isoflavins, caffeine, cyanogenic glycosides, methyl, all these things are naturally present. We are exposed. So essentially, it doesn't mean that we should probably look for, we need to look for these things, but as long as the concentration is de minimis and we know the source, we can actually manage. And intentionally added food, which is the modern industrial world, the food additives world, will come to very, very important in this modern world. As the population increases, the area of the agricultural land is diminishing. We need to think of creative ways of providing quality food at a reasonable cost to the 7 billion people that we are here. So new food ingredients, example, genetically modified. I know the controversy, but I'm talking strictly as scientists here. Genetically modified crops have been tested and tested and tested for last 25 years. And it is, in fact, 75% or 80% of the American crops is nothing but genetically modified. So essentially, unless we actually take advantage of the genetically modified crops, which decreases the amount of pesticides to be used, it is foolhardy in the modern world of any country not to use it, take advantage as the population is increasing to provide quality life, quality food. That's very important. And functional ingredients, obviously, we had some examples. So melamine we talked, and Ayurveda adulterants. That's almost many of us take in India for granted. Ayurvedic medicine, take a bottle, take it. So we have no idea of the composition of it. And I myself, let me tell you, I'm one of the greatest users of Ayurvedic medicines. It increases life expectancy. Sorry for that. Okay, unintentionally added. Here is the dioxins, PCBs, acrylamide. Many of these things are with us, and they're going to be with us for a long time. But remember, 25, 30 years ago, when I started my career in Dow Chemical, as soon as I entered, we found an incident called a compound called dibromochloropropanone. How many of you raise hand? Dr. Kanunga would know that. There's a nematicide used. It produces fertility. I had to go around the companies and talk to the employees what this means. What happened is when we did the investigation, the DBCP, dibromochloropropanone, was used for 20 years. Suddenly, one change of a shift of manager, supervisor, he did not implement PPEs, personal protection equipment. People were actually drenched in that compound. 23 employees of one of our company, 3 other companies were there. They were sterile, zero sperm. So it is not the property of the chemical. It is how we manage with the chemical makes a big difference. Okay, now endogenous toxins, these are simply, I don't want to read all these things. These are toxins that are present in plant foods. Whether you are a vegetarian or whether non-vegetarian, you are guaranteed to get some or parts of it depending upon the type of the source that we are eating. Natural contaminants, obviously edible plants, toxic plants, contaminants, and microbial toxins we touch base. Now, Dr. Kanunga, again, he very succinctly put it in 3 sentences where he covered all these things. Packaging materials, BHA, butylated, PCPs, polymers, pentachlorophenol, metals, processing chemicals, PAH from cooking, solvents, environmental compounds. This is very important. Natural and anthropogenic compounds are with us, and they're going to be there for time immemorial and forever. So essentially, we need to understand, manage lead, mercury, of course, in addition to fumigants and pesticides, which are newer ones. Now, what is food safety? Food safety, the ways food may become a source of illness, foodborne diseases, we talked already this morning. Food hazards may have to do with pathogens. This is just a summary of what I talked last 4 or 5 slides. Food pathogens, contaminants, pesticides, food additives, natural and nutritional imbalance. That's one. Okay, now let's get, this is simply a kind of a types of food additives that you and I are consuming in different quantities, proportions, different countries, different regions of the country, and different seasons of the country. So the US Food and Drug Administration has categorized the food additives, which are into 6 major categories, which are preservatives. I'm giving major categories, but in each of the categories, or each of the antimicrobial, there are dozens and dozens of them. They're all approved, don't misunderstand. Antimicrobials, anti-browning, and antioxidant. These are very, very crucial for food industry, food technology. Textures, stabilizers, emulsifiers, these are absolutely critical, but they're in usually larger quantities. Okay, then flavors. Obviously, all of us, human beings, we want nice flavors. There's no question. That's why we enjoy food. And foods and sweeteners and natural synthetic flavors are there. Miscellaneous enzymes, catalysts, solvents, propellants, these are all things that are used in various industries. They are essential for the modern life, no question. And nutritional vitamins and coloring. Coloring agents, even though they're very tiny fraction, there was a lot of controversy of colors back in the 60s because some of the coloring agents, their family come from the carcinogens, not the colors itself. So they had a history of that. So now that has been proved and that these don't cause any carcinogenicity. As a result, there are a limited number of approved colors very widely used. Okay, now scope of food additives. Just to give you a statistics, this is approximately there are 3,000 food additives that are currently approved. I want to say approved in the market. 1,800 of the 1,800, more than half of the additives are flavors, flavoring agents, 1,800. Texturing agents are used in the highest quantity because of the volume bulk involved. Soft drinks are biggest market for these things. The 4 direct additives contributes 93% of the total additives used. What are those? You know, they're common things. Sucrose, salt, corn syrup, dextrose. None of us can get scared with this, 93% of it. So we are talking of these are things essential that we consume all the time. Some of them are part of our body. So in EU, very important, I made a note, sucrose and salt are not classified as food additives, just a footnote. Now, problems associated with food additives of safety. What are the problems with food additives, food safety? Absolute safety cannot be proven with anything. There is always a finite chance. That's where this threshold comes into the picture. My future next speakers will talk about. No protocol is adequate for all compounds. There is one thing is there, a lot of us look for guidance, look for something. One of the uniqueness of the Food and Drug Administration is they have general guidances, documents, things that is simply a starting material, that is not a regulation. Each of the issues that come up with, we should come up with the unique design of testing those materials, use all the technology, science, biochemistry, knowledge, toxicology, and use that. The another issue is extrapolation from high dose to low dose. By definition, we toxicologists are forced, and in fact, it is a good idea to do the highest dose must induce significant toxicity to animals. Because you cannot do a toxicity test with 1 milligram, 5 milligram, 10 milligram with the food additives and say, hey, nothing happened. When I first started to studies and carcinogenic studies with sweetener, we went to the highest quantity the animals can tolerate, which is 2 grams per kilogram of the food additive given there, a lot of material. So in that design mode, the toxicology studies, you automatically get some effects at the high dose. The reason we want to is we are always trying to for using only a small number of animals, 10, 20, maximum 50 animals or 60 animals per group, per sex. So we are going to use this compound to billions of human beings across the world. So there will always individuals who are sensitive. Not that this design will pick up every sensitive individual. At least it can say that given certain sensitive individuals and certain exposure, it can have this particular effect. So, and finally, extrapolation from animal data to human beings. This is almost an age-old phenomenon, but certainly in the last 50 to 80 years that the toxicology testing has been in existence, we learned a lot of lessons. We learned what animal data means. And there are some unique things that happen in animal studies, liver carcinoma problems. We have actually resolved those. These are unique for animals, not relevant to human beings. Vice versa, there are certain things occur in humans we cannot detect in animals. So we have the examples. Genetic variation we talked this morning, high cost obviously testing. Now whole foods. Whole food is a complex mixture, wide variation in composition, and impact on dietary imbalance. Large quantities cannot be given to animals. It produces an imbalance. The imbalance itself will produce toxic effects, which has no relationship to the quantity of the diet itself. Now, safety consideration. Foods to which substance is added, we need to test. Levels used in such foods, very important, what levels are going to be used. Always a toxicologist, I know very few are toxicologists here. As a toxicologist, we should not start, clients do come there, hey, can you start Monday morning study? Absolutely not. You need to understand the issue, understand the problem, think of it, have a plan and design, and then explain this is what the design is based on the problems you pose to him. If these assumptions do change, then our toxicology studies design changes. And purpose for which the substance is used, very important to consider, in particular for food additives. And more important, the population. If we know something is going to the only baby foods, certainly we need to take into consideration pregnant women or geriatric individuals. Okay, how is safety assured? Now, we come to the regular, now how safety is assured. Let me tell you, we have very, very good systems in place in the field of animal toxicology, preclinical toxicology, where we can, I would say, practically 90 to 95% of the things that occur in human beings we can detect based on the designs we have. So it is probably maybe overarching regulation of law requiring manufacturer to ensure that the food be safe and the additives are safe. No question, every major manufacturer that is there conducts every test that is required, and no regulatory agencies, particularly the Western world which marketed there, they have very stringent requirements. FDA does not get a gold medal if they approve a product, absolutely not. So they do extremely thorough, likewise the Europeans, likewise the Japanese, other things. And fortunately in India we use the systems that are in place, and certainly I can assure you that anything that is marketed here by reputable companies, absolutely it is safe, no problems at all. But the biggest problem if some of these things happen is the abuse. Is the concept of 2 tablets works, let's take 4 tablets. That kind of mentality is the one which is actually creating occasional these incidents that becomes the headline. It is not the compound per se under use conditions. Okay, now another concept which I want to move, which is the GRAS. Many of you know it, generally recognized as safe, because this happened back in 1958 when the Delaney clause came into existence. They told, you know, existing products, obviously we cannot test to the Delaney clause because these have been used for age-old for generations, at least a couple of generations. So they made that as any compound that is already in existence as of 1958, it is considered as generally recognized as safe. Subsequently, even though there are some compounds which are there, we can be exposed, then they say let us classify them into GRAS based on some testing or some risk assessment. So what I'm going to, again, the section 406 of the FDC Act came into existence for that. The next slide, which sort of depicts what these systems are in place. These food additives, sorry, yeah, data, the exclusive, this is the food additives, the ones where exhaustive testing happens. Data is exclusively generated by the primary manufacturers, reviewed and approved by the FDA, takes years and years to come up with the food additive. Aspartame, which I worked intensely, it took several years to come on the market, then off the market, it came on the market, a lot of history. I had a case study, but we decided not to present that. So evidence of safety requires full testing, no question. Going to the GRAS substance here, limited data exclusivity. That means if suppose one very well known already, you can have a generally recognition by experts like our audience or panel members. Any one of the panel members, including me, can write a document which I have written and submit to the FDA. In my judgment, based on the literature survey, we don't necessarily have to do the testing ourselves. And obviously, FDA has a chance to review, so that's accepted. Now, there are some things that new additives that come up which are not known at all before. The primary manufacturers can actually do the testing, go to the FDA, get it as a generally recognized, so that wide spectrum of industries can take advantage of it. This is the second class, which is reviewed and approved by the FDA, and that's widely known consensus. Evidence of safety requires full testing. Now, just one slide before that, 2 ladies were having a conversation. I don't know, I'm sorry. You may not be able to read the bottom. One lady said, you know, I take only organic food. You know, it's very fashionable to say organic food, and I would not touch an organic food because you don't know what kind of pests and other things that are there. The other lady on the other side, who is a little older, said, I don't eat, at my age, I can use all the preservatives I want, and she's already 95, doesn't matter. So the bottom line is, you know, all the food additives or anything that comes up, particularly the added food additives, they have been absolutely unconditionally tested. Only in these cases, isolated cases, yes, the misuse has produced problems, and extraneous cases where certain populations do respond. There is nothing, no animal test can detect. Those are isolated, kind of, again, 10 to the power 6, 1 in a million. That can happen, it has happened, and certainly we need to be aware of. Most ethical companies actually come to the rescue, take precaution, immediate actions, response to that. So with that, I want to conclude my talk. Thank you very much.